For centuries, we built entire civilizations on a single assumption. We looked at skin color, facial structure, the shape of eyes, and the curl of hair, and we said, these differences mean something deep, something fundamental, something written into the very code of human life. Empires rose and fell on that idea. Wars were fought over it. Laws were written to enforce it. Textbooks taught it as settled truth, and generation after generation absorbed it without question. Then, scientists did something no one had ever done before at scale. They didn't look at skin. They didn't measure skulls or map facial features. They went straight to the source, the DNA itself. They collected genetic material from populations across every continent on Earth, from the mountains of East Asia to the forests of Central Africa, from the Arctic tundra to the islands of the Pacific. They compared billions of genetic markers across thousands of individuals, representing the full breadth of what we call human racial diversity. And what they found didn't just challenge the assumption, it dismantled it completely. The results were so unexpected, so structurally different from everything the world had been told, that researchers had to sit with them before they could even begin to explain them. Because the story written in our DNA is not the story we thought we knew. And once you hear it, you cannot unhear it. To understand why the findings were so disorienting, you have to understand how deeply the concept of biological race had been embedded in scientific thought. It wasn't just a social attitude. For most of Western history, it was considered empirical. In the 18th and 19th centuries, some of the most respected scientists in Europe built elaborate systems of human classification. They named and ranked what they called distinct biological races, assigned characteristics to each, and presented these categories as natural facts, as observable and measurable as the distance between stars. The logic seemed clean on the surface. Different groups of people looked different. They lived in different parts of the world. They had different customs, different languages, different physical traits passed reliably from parent to child. Surely, the thinking went, these visible differences reflected deeper biological divisions. Surely the human species was divided into distinct biological lineages as separate and defined as breeds of domestic animals. This framework became the foundation for medical practice, legal systems, and social policy on a global scale. Even in more cautious scientific circles of the 20th century, the broad assumption persisted that the visible differences between human populations reflected something structurally significant in their genetic makeup. That races, however defined, were biologically real in a meaningful sense. That the categories we use to sort people were rooted in something deeper than culture or geography. That is what was waiting to be tested. And that is what the DNA revealed. The ability to actually test this idea at the genetic level only became possible in the late 20th century, and it accelerated dramatically in the early 21st. The Human Genome Project, completed in 2003, gave scientists their first complete map of human DNA. But the real revolution came when sequencing technology became fast and cheap enough to analyze not just one genome, but thousands of them simultaneously. Researchers began collecting genetic samples from populations worldwide, and the datasets grew into something unprecedented. Projects like the International HapMap Project, the 1000 Genomes Project, and dozens of subsequent studies built libraries of human genetic variation that spanned the globe. For the first time in history, scientists could compare the genetic blueprints of populations from across all of humanity, not just theorize about them. What they were looking for was the pattern of genetic variation. Where were the differences concentrated? How sharply did one population's genetic profile end and another's begin? Were the commonly used racial categories reflected in the genetic data? Did the boundaries we draw between races correspond to actual boundaries in the DNA? The tools were precise. The data was real. And the answers that came back were nothing like the assumption that had stood for 300 years. Here is the finding that restructured everything. The genetic difference between any two individuals from what we consider different races is, on average, barely larger than the genetic difference between two individuals from the same race. Let that sink in. If you take two people we would visually categorize as belonging to completely different races, one from sub-Saharan Africa and one from Northern Europe, their genomes are roughly 99.9% identical. And the tiny fraction that differs? The vast majority of that variation exists within populations, not between them. Scientists found that approximately 85 to 90% of all human genetic variation occurs between individuals within any single racial group. Only 10 to 15% of variation exists between the groups themselves. This was not a minor revision. This was a structural overhaul. The data was saying something unambiguous. The boundaries between what we call races are not written into the genome the way anyone expected. There is no clean genetic divide, no separate lineage, no discrete biological grouping that maps onto the racial categories humans have used for centuries. But the story gets more layered from here, because the DNA didn't just show the absence of deep separation. It also showed something remarkable about presence, specifically the presence of shared ancestry. When researchers began tracing the genetic relationships between modern human populations, a consistent picture emerged. All living humans trace their ancestry back to a relatively small ancestral population in Africa. The further you go back in the DNA, the more every human being on Earth converges on the same origin point. The diversity within African populations is actually greater than the diversity between all non-African populations combined. This single fact turned the old model completely upside down. The genetic data places Africa not at one end of a spectrum of human diversity, but at the center of it. Every population outside Africa carries only a subset of the genetic variation that exists within Africa, because all non-African populations are descended from groups that migrated out of the continent, bringing only a portion of the original genetic diversity with them. At this point, a reasonable person asks the obvious question: if race isn't written into our DNA the way we thought, then why do people look so different? Why does skin color, hair texture, and facial structure vary so dramatically across populations? The answer is both scientifically fascinating and philosophically important. The visible traits we associate with race, the traits that have driven so much of human history, are controlled by a remarkably small number of genes. Skin color, for example, which is perhaps the most socially weighted physical characteristic in human history, is determined by a handful of genes out of the roughly 20,000 that make up the human genome. These traits changed relatively quickly in evolutionary terms because they were under strong environmental pressure. Populations living near the equator developed darker skin as protection against intense UV radiation. Populations in northern latitudes evolved lighter skin to facilitate vitamin D synthesis in lower sunlight. Eye shape, hair texture, body proportions, many of these characteristics reflect adaptations to specific environments, shaped over thousands of years by natural selection. What this means is that the traits we use to define race are among the least representative of our total genetic difference. We built a global taxonomy of human beings on the features that changed the fastest and the most superficially, while ignoring the vast landscape of genetic similarity that lies beneath them. We looked at the most visible layer of human biology and mistook it for the deepest one. It was like judging the fundamental composition of two buildings by their paint color while ignoring that they were made from the same architectural blueprint, the same materials, the same structural design. The implications of this data reach far beyond laboratory walls. They reach into medicine, into identity, into politics, and into the way human beings understand themselves. In medicine, the reclassification of human genetic variation has already forced significant changes. For decades, medical research calibrated certain treatments and diagnostic standards along racial lines, assuming that biological differences between races were significant enough to justify different clinical approaches. The genetic data has pushed medicine toward a more sophisticated model, one that looks at individual genetic profiles and specific ancestry markers rather than broad racial categories. The result is more precise, more personalized, and more equitable medical care. Diseases once thought to be racially specific are now understood as genetic variants that exist at different frequencies in different ancestral populations, which is a very different thing. In forensics and population genetics, the findings have opened new methods for tracing ancestry with extraordinary precision. By analyzing specific patterns of genetic variation, researchers can now identify not just continental ancestry, but regional ancestry, sometimes narrowing someone's genetic heritage to a specific historical migration corridor thousands of years old. This technology has rewritten our understanding of ancient human movement, showing that prehistoric populations mixed and migrated in ways that make the modern concept of bounded racial groups look almost fictional by comparison. And in the broader cultural conversation, the one that has been ongoing for decades around race, inequality, and identity, the genetics data provides something essential: a scientific foundation for separating biological reality from social construction. The data does not say that human populations are genetically identical. It does not erase the real differences in disease risk, ancestry, or genetic diversity that exist between populations. What it does say is that the system of classification humans built around race does not reflect what is actually happening in the genome. The categories are real as social facts. They are not real as fixed biological divisions. Zoom out even further, and the genetic record reveals something almost philosophical. The human species is, by the standards of biology, extraordinarily young. We have been on this planet for roughly 300,000 years, a blink compared to the millions of years other primate species have had to diverge and differentiate. Our nearest evolutionary relatives, chimpanzees, show significantly more genetic variation within their species than humans do across the entire globe. We are, in genetic terms, a remarkably homogeneous species wearing an impressively varied surface. Every population of humans on Earth carries within its DNA the fingerprints of movement, of encounter, of mixing. There is no population anywhere that has existed in perfect genetic isolation. The San people of southern Africa, often described as among the oldest continuous populations on Earth, shared genetic markers with populations thousands of miles away. The indigenous peoples of the Americas carry genetic signatures linking them to ancient migrations from Asia. The populations of Europe are the product of at least 3 major waves of ancient migration, each one rewriting the genetic landscape of the continent. Even within what we call races, the mixing goes back far deeper than recorded history. The picture that emerges from the full body of genetic research is not a tree with separate branches. It is something closer to a river delta, branching and braiding, splitting and rejoining, flowing outward from a single ancient source. Every human alive today is the product of that flow. Every genome is a layered record of every encounter, every journey, every generation that came before. There is a version of this story that ends simply: science has spoken, the old categories are wrong, case closed. But the more honest version of this ending is more complicated and more demanding. Because knowledge alone does not change the world. The genetic data has been available and increasingly clear for decades, and yet race continues to function as a powerful, consequential, real social force in nearly every society on Earth. The fact that racial categories are not sharp biological divisions does not make them disappear. People are still treated differently based on how they look. Communities are still shaped by the legacies of racial classification. Identities are still formed around racial belonging, often in ways that are deeply meaningful and culturally rich. What the science does is change the ground beneath the arguments. It removes the biological bedrock that was used to justify hierarchies, to enshrine inequalities, to declare that some human beings were fundamentally different in kind. It forces a confrontation with the question, if we built this structure on a foundation that doesn't exist, what does that mean for the structure itself? It also offers something quietly extraordinary. The same Dina that dismantled the myth of biological race also revealed the full depth of human shared ancestry. Every person alive today is connected, not in metaphor, but in molecular fact, to every other person alive. The same blood that runs through the most distant population on Earth ran through common ancestors not so many thousands of years ago. The human family is not a poetic sentiment. It is a genetic reality. Scientists compared the Dina of every race on Earth. And what they found was not the sharp lines of separation that history had assumed. What they found, beneath all the surface variation, beneath the skin colors and the eye shapes and the geography, was something that has always been true and has never stopped. Being astonishing. They found us, one species, one origin, one story, written in the same four-letter alphabet across every genome on the planet. The results didn't just shock the scientists, they redrew the human story from the